Right, we're now moving on to some food technology aspects of lipids. Most of these terms we were familiar in the context of a range of foods containing fats. Maybe not across rheology. Rheology is a study of the flow of matter, often liquids, but often soft solids, gels and pastes, which are often very relevant to foods containing a lot of lipids. And there's a couple of examples. I guess maybe not the caramelised toffees so much, but certainly spreadables of all sorts. Taken together, these factors, and it is an influence of all the factors acting together, these factors influence the characteristics of a particular food in often complicated ways. We'll have a look at chocolate as an example of one of the more well understood examples. Yeah, the melting points of triglycerides are a function of the length of their carbon chains of their fatty acids, as we discussed before. The bigger the molecular weight is, the more quickly something will crystallise. However, lipids are very diverse in structure. This may produce, for example, a wide range of melting points. It may seem a bit little odd to refer to this process as crystallisation, which we normally associate with very regular structures, such as sodium chloride crystals. Here it refers to a structure that is more regular than just an amorphous collection of molecules. A similar situation is discussed when we talk about starch. Yeah, so unsaturation again is another key factor. The saturated triglycerides on the left can pack more closely together than the unsaturated ones on the right. So the symmetrical saturated ones are easier to pack and need less energy, whereas the asymmetrical ones are harder to pack and take up more energy. You see quite a lot of this in the literature, where it's discussed in actually quite general terms. The model of stacked chairs is often used to describe the packing of TGs. Triglycerides. Chairs can be stacked in a number of ways, including so-called double and triple packing, as shown there. And that's obviously just a random packaging of chairs. And you'll see, as I mentioned, you'll see very similar images of triglyceride packing in textbooks and papers. Like stacking chairs, the analogy soon breaks down and the chairs fall over. Yeah, so. Okay, so polymorphism. This is an important factor. This is when a solid can exist in a number of different crystalline forms. The main categories we're interested in often describes alpha, beta prime, and beta. So that's beta prime, one with a little asterisk next to a little comma next to it. There are many other systems in use, it has to be said, including within food. So I've just picked this one, but you may open textbooks or papers which will use a somewhat different system. The thing to notice here is the overall change in characteristics associated with the different forms. So smaller crystals, low melting points, high crystals, high melting points, and the more stable. Yeah, so the small crystals seen in alpha are unstable and convert spontaneously to beta and beta prime, often with a significant influence on food structure, maybe producing large crystals in a grainy texture and perhaps flakiness. Yeah, so it's unstable and will convert to beta prime and beta. So chocolate. The raw cocoa bean contains a high proportion of lipids, with carbohydrates as the runner-up. Cocoa butter, often called theobromine oil, is a pale yellow edible vegetable fat extracted from the cocoa bean. It has a high proportion of saturated fats and melts at around normal human body temperature, although there's quite a range, as you can imagine, given the different fatty acid composition. Cocoa butter has about 6 polymorphs. You may again see other ways of describing them. There's a lot of debate about whether they're in fact crystal polymorphs, but we'll work on this basis. Form 5 is the highest quality chocolate with good texture, taste, glossiness and snap. So you would see this, for example, in Belgian chocolate. And as I said, the melting point is about the melting point of our mouth, which is about 34 degrees. Yeah, many years ago, the product Treats, down here, was marketed as melting in your mouth, not in your hands. This is probably more due to the coating, which is a sugar coating basically, rather than quality of chocolate. If you're interested, see the video for marketing back in the day. Yeah, this video is a good review of the industrial process, this video up here. I'm not going to go through it here, the details are not examinable, the underlying concepts may be. So have a look at that in your own time. Okay, Conching. Chocolatfabriken, Lindt and Sprungel AG, more commonly known as Lindt, is a Swiss chocolatier and confectionery company founded in 1845 by Rudolf Lindt. Before old Rudolf, chocolate was hard and gritty and rather bitter. He developed the Conching process. The original equipment looked like a conch shell. Essentially here, repeated mixing reduces particle size, leading to increased smoothness and higher quality chocolate, which is often in the 5 polymorph. And there's one of the original machines. A bit more sophisticated these days, but here's the chap who developed this. Yeah, so tempering. Tempering is a controlled melting and cooling cycle designed to increase the proportion of the most desirable polymorph, that is to say 5. And there's a summary of the process. We'd like a tempering machine for the food lab. Okay, so a couple of videos on tempering here. The first here shows you how to do this by hand, and the second shows a short, small automatic machine. Here's British chocolate. Chocolate bloom is either of two types: a whitish coating appearance surface of chocolate, fat bloom caused by changes in fat crystals in the chocolate, and sugar bloom due to crystals from the action of moisture on the sugar. Fat and sugar bloom damage the appearance of chocolate and limits its shelf life from an appearance point of view, though they don't have significant sort of food safety issues associated with them. Right, so there we are. That completes lipids. Thanks for listening.